start.
All right. Hello, everyone. I'm Ishita. Nice to meet you again. So I'm just going to share my screen here. Today, I'm going to be talking about, everyone can see this?
Yes.
Yes? Okay. So today I'm going to be talking about the technological revolution in medicine. And I know that's very general. It's because I was finding it really hard to pick one or two specific topics to talk about because there's so much going on in the sphere that's under the umbrella of this class. So I chose two things that are very exciting. They're very different, and I think they're also will interest you, hopefully, as much as they interest me. So the two things I'm going to be talking about are gene editing, and then also the idea of consciousness and what makes consciousness. So starting with genetics. So we have talked about a little bit about regenerative medicine through Dr. Soles's lectures. So basically, regenerative medicine is the process of using stem cells to replace damaged tissue. So this is something that is currently being used in clinical applications, and it can be used in basically any condition or disease where a certain type of cell is required, but is not being produced in that patient. So for example, if a patient has macular degeneration, we could use regenerative medicine to create retinal cells to help not prevent, but to help improve upon the condition. And similarly in diabetes with insulin producing cells and Parkinson's with dopamine producing cells. So this is also in Dr. Soles's lecture, he mentioned regenerative medicine with entire organs. So this is used in both specific cell types and in organs. Some other interesting applications can be using stem cells themselves to transplant into a patient. So in diseases like leukemia, where the bone marrow is the diseased part and also the part that creates stem cells, that entire bone marrow can be replaced using stem cell transplant. So I just want to introduce a few different studies to kind of give more concrete examples of what I'm going to be talking about. So the first one that I want to talk about is Dolly the Sheep. Has anyone here heard of Dolly the Sheep? Yeah? Okay, yeah, she was born around the same time as a lot of us in 1996, and she was pretty groundbreaking in 1996 because Dolly the Sheep was the first cloned mammal ever, successfully cloned. So she lived to be almost 7 years old, and she died of natural causes. So she died of a lung infection that had nothing to do with the cloning process itself. So all in all, this experiment was quite a success. So to explain how they did this, how they cloned this animal, basically Dolly had 3 mothers. Can you guys see my mouse right now? Yeah, okay. So Dolly had 3 mothers. She had one, this one right here, donated an egg. The other one donated her nuclear matter. So this was the sheep that was genetically cloned. And then the third one was the surrogate sheep, which they implanted the blastocyst into, and she's the one that carried Dolly to term. So this was, although it was a huge deal in 1996, people were concerned about how this could lead to human cloning, which obviously now fewer people are talking about. No one's really concerned about it. But after 1996, animal cloning actually became pretty commonplace. So in 2008, the US legalized cow, goat, and pig cloning. because there wasn't enough of a difference between uncloned and cloned animals that there was any reason to stop them from cloning. So they are cloned for agriculture now pretty frequently. So it's not really a big deal anymore. But some issues with this, with embryonic stem cells in particular, is that there is limited accessibility of these embryonic stem cells. There's the possibility of immune rejection because there were 3 donors used in this Dolly the Sheep cloning experiment. So any one of these could have rejected the transfer. And then it's also ethically complicated, especially when we begin to talk about human embryonic stem cells. Obviously, lots of people have lots of different opinions on what embryonic cells can be used for, and if they can be, messed with for science. So I also forgot to mention, but you guys can feel free to unmute yourselves and ask any questions or any comments. Feel free to chime in whenever. So some more controversies regarding embryonic stem cells. Obviously, people are concerned if human cloning is the next step. It doesn't seem like it right now. And when we're talking about humans, obtaining eggs requires ovarian stimulation or heavy hormone doses for hyperovulation. So embryonic stem cells are isolated from humans. Like this is something that happens currently, but it also has, we're not sure of the lasting issues that can be caused by these heavy hormone doses in females that are required to isolate large amounts of eggs. Another question, why is selling most body parts illegal, but not eggs, even if it is for science? And should clone embryos have the same moral status as normal embryos? So this would apply to humans, but I think it also applies to animals as well. So the animals, cattle animals that are being bred for agriculture, there are strict humane rules that apply to animals that are bred for meat. Do the same rules apply for cloned animals? Are they their own animal is kind of the main moral question that a lot of people have trouble answering. So does anyone have thoughts on any of this so far? Comments, questions?
I had no idea that the cloning of sheep is now used often. Thank you for sharing that. Also, what do you mean as moral status? Uh, do you mean that a person that is cloned would have a different standing in the way we apply law to them, or?
Yeah, that's basically the question. Because we apply a lot of value to human embryos, right? And would that value remain undiminished to embryos that have been genetically modified or that are cloned? Do we consider clones their own separate entity, or are they okay to be used for, scientific experiments or to be slaughtered for meat, and they don't have to deal with as strict laws because they're just clones?
But
So
then, do you think that we would go into a stage in time in which these clones grow, and then some of them become lawyers, and then they create their own laws, maybe? Because imagine that state in time, while they develop their, potentially they would have the same brain capacity and the same thinking abilities. That stage in time would be worrying because uh, we could use them, as you said, to slaughter them to get the organs that someone else needs. But is there value in their life? I don't know. This is very interesting to me. I don't know what others think about that.
Does anyone else have any thoughts about this?
That is actually a book. It's called Never Let Me Go, where they make clones and they harvest them for body parts. So yeah, if you wanna.
I think it's really, can you guys hear me?
Yep.
Oh, okay. I think it's really interesting what you brought up, Eduardo, about like kind of these clones having their own laws and that sort of thing. I think that's a really interesting idea. One thing that I might add to kind of a secondary discussion is that third point, why is selling mouse body parts illegal but not eggs? When I think about that question, I kind of think back to sperm cell or like sperm donors. So why is that so easily accepted, but not eggs? So that's just something I thought of.
Yeah, I think the laws surrounding that are really interesting. I like the reason that sperm donation is so much more, easily accessible is just because how minimally invasive it is. It's much easier. You don't really have to go through any kind of procedure. But yeah, but at the same time, if you donate sperm, you get paid a fraction of the money that you get if you donate your eggs. You get, I'm not sure, like several thousands of dollars if you donate eggs. So it is really interesting to think about. Does anyone have any other comments before I move on? All right, I'll keep going, but I have several slides that just have questions on them, so we can stop on those and just talk things through. So yeah, so similar to the controversies faced by the use of embryonic stem cells, I wanted to, sorry. Oh, this slide isn't working. Just a second, I'll stop and restart the share. All right, I will just show you this one slide, not in presenter view, but basically it was just a slide that had a whole bunch of headlines, newspaper headlines about how controversial IVF babies were when they were first introduced in 1978. People were concerned. There were lots of moral objections from the public saying that if a couple is unable to conceive the natural way, then should they be allowed to, have babies through an unnatural way, as it was called. But now, obviously, even though it's only been 42 years since that first IVF baby, we don't really care anymore. Like 4 million babies in the US are born from IVF every year, which is a huge amount. And no one really cares. You're actually seen as socially incorrect if you disagree with IVF babies. So I like to bring this up just because, oh no, it's working, that's nice. I like to bring this up just because we talk in this class so much about exponential change, and these things occur in clinical practice as well. Like this is a huge change in just the span of 40 years. So the less controversial way of using stem cells that was recently developed are called induced pluripotent stem cells. This is probably something that you guys have heard about because it's such a huge development. The creators of this, Takahashi and Yamanaka, they won a Nobel Prize for coming up with this technique. So basically, to create induced pluripotent stem cells, you can use any fully differentiated adult cell. So you can use skin cells, you can use urine cells, blood cells. It doesn't have to be invasive at all. You can just give like a skin sample or a urine sample. And these scientists found a way to isolate through several steps 4 specific genes that most resembled embryonic stem cells. And after culturing them for several months and isolating them, they were able to create fully pluripotent cells from these adult cells. So basically, this was pretty groundbreaking. I still think it's amazing that adult cells can be brought back to their immature state, and they can be redifferentiated into whatever we want them to be. So of course, everything has advantages and disadvantages. So like I said, an advantage that is pretty significant is that it's minimally invasive. You can just give a blood sample, which is really easy to do, and create a culture of your own stem cells. It's suitable for autotransplantation with low risk of rejection because theoretically, because the cells are being made from your own adult cells, it should be a perfect match for your genetic makeup. And it's also great for disease modeling in vitro with no risk to the patient. So if you culture these cells and create a sample in a petri dish of a specific patient's cells, you can apply certain therapies or medicines in that petri dish and see how that person's cells would respond to it without having to deal with the patient going through the process of trying one thing for a few months. If it doesn't work, they try another thing for a few months and deal with the side effects. So some disadvantages are that the time between the extraction of the cells and transplantation is quite long. So there's always a risk of transferring mutations to the patient. So just through spontaneous mutations, because it takes months to culture these cells, and also through the fact that we are applying external factors to them to get them back to stem cell, and then redifferentiating them into what we want it to be. And while the entire idea of induced pluripotent stem cells are based on getting them as similar as possible to embryonic stem cells, they aren't embryonic stem cells. There are always going to be things that we don't know, always going to be genetic and environmental factors that we can't control. Like we'll never have, I guess I can't say never in this course, but we don't have full control of what these cells are differentiating to. So the next thing that's related to gene editing, and this is just background for the next study I'm going to talk about, is CRISPR-Cas9. And I think that probably a good amount of you are familiar with this. This is a gene editing system, and the 2 scientists responsible for creating it were recently awarded the Nobel Prize in Chemistry just this year. So this is a pretty recently developed technique. It uses a modified bacterial protein with RNA markers, and the Cas9 enzyme induces double-strand breaks in the DNA. So if this video in the corner is working, you'll be able to see that. And CRISPR is a protein that makes transcription factors that target specific genes. So using this system, scientists can rewrite to either activate or silence or in any way target a specific gene and reinsert it. You can also take out the existing gene and insert a completely new gene wherever you want it to go. So it's quite valuable as a tool to explore what we can do with gene editing. Okay, so this study is really interesting to talk about morally and ethically. So if you guys are familiar with the gene edited babies that were born a few years ago, yeah. So Dr. He in China, he wanted to create genetically edited babies that were resistant to HIV. So I'll just explain the study to you a bit, and then we can talk about the ethical considerations. So in his study, out of the couples that he was working with, the men had HIV and the women did not. So basically he reasoned that the whole point of this study was that so that the babies could be born without HIV. Also, something interesting to note, all the male participants' HIV was well controlled by current treatments. There are also current treatments that allow parents with HIV to give birth to HIV-free babies. So I just wanted to make that clear that this wasn't at all necessary. These couples could have had babies without HIV and without going through gene editing. So interestingly, he gave official notice of his work on November 8th, 2018, after twins were born from this study. So this work had been going on for at least a year before he actually gave notice of his work. He also had misleading consent forms that called the project an AIDS vaccine development program. And also interestingly, he promised insurance coverage for the children conceived through the project until they're 18. So yeah, I won't say anything. I'll let you watch the video and decide for yourself. Last thing, he also forged ethical review papers. So just kind of, yeah, just setting the scene, but I do want to share with you a quick video of him because I think it just helps figure out what his, like where he's coming from, what his mindset is. So I know that this can have issues with audio, so let me know if you can't hear it.
Two beautiful little Chinese girls named Lulu and Nana came crying into the world as healthy as any other babies a few weeks ago. The girls are home now with their mom, Grace, and the dad, Mark. Grace started her pregnancy by regular IVF with one difference. Right after we sent her husband's sperm into her egg, we're also sending a little bit of protein and instruction for a gene surgery. When Lulu and Nana was just a single cell, this surgery removed the doorway through which HIV entered to infect people. A few days later, before returning Lulu and Nana to Grace's womb, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again deep sequencing Lulu and Nana's whole genome. This verified the gene surgery worked safely. No gene was changed except the one to prevent HIV infection. The girls are safe, healthy as any other babies. When Mark saw her daughter for the first time, he said that he never thought he could be a father. No, has found a reason to live, a reason to work, a purpose. You see, Mark has HIV. Discrimination in many developing countries make the virus worse. Employer fire people like Mark doctor deny medical care, and even falsely sterilize a woman. Mark and Grace couldn't bear to bring a child into that world of fear. Mark's words told me something I didn't fully appreciate. A gene surgery that could save a child from a lethal genetic disease like cystic fibrosis or from an life-threatening infection like HIV. It doesn't just give that little boy or girl an equal chance at a healthy life. We heal a whole family.
Okay, I'm gonna stop it right there because I feel like that gave a pretty good understanding of what he thinks of his study. He continues to defend it, which is not surprising. So I just wanted to show you that because a lot of the time from scientists' perspective, or maybe from your perspective as well, you see, if you saw this kind of study, you might think, obviously that's unethical, like we're not medically there yet to be able to gene edit kids like humans, and for that to be okay. But this is still a debate because videos like that, like the one he has put out, are, you know, emotional and convincing in that a lot of people, a lot of the public opinion in public forums and stuff, people are still debating if this is the worst thing. They're like, maybe it can be a good thing. People with HIV are saying, this is great, like no one should have to suffer like through the things that I've suffered through. So this isn't necessarily a black or white thing that we might think it is, or that I previously thought it was before actually reading what the public opinions about this are. So if you're wondering what happened to the babies, they were born in late 2018, but because them and their parents are completely anonymous, basically we have no information on them at all. So his research notes were actually released in excerpts earlier this year. And there were some pretty concerning things inside them. And I think that the video he released was very misleading. So I just wanted to talk about that a bit. So what he was targeting was this Ccr5 mutation. So Ccr5 is the gene this mutation, it exists within the normal population, and it provides resistance naturally to some parts of the population. So there are people who have this mutation and are naturally resistant to HIV. And that's what he was trying to imitate in these embryos. So unfortunately, through his research notes, we found that he modified genes close to that mutation, but it's not confirmed that it was exactly there. We also see from his notes that there is evidence for mosaicism. So some cells in the embryos did get that mutation that they were going for, but some didn't, which would defeat the entire purpose if there are some cells still susceptible to HIV. While he said in his video that everything, they tested the embryos and everything was perfect, their notes reported one off-target mutation that they found by testing some sample cells from the embryo. And this would not encompass the whole embryo because they only tested a few cells. So we don't actually know how many off-target mutations there were. And Dr. He himself was charged for 3 years in prison and fined $430,000 US. He pled guilty, but he still defends his research. So obviously this brings up a lot of questions. Wait, I'm gonna see what I have on the next slide. Okay, yeah. So does anyone have any thoughts or questions about this study in particular?
I would like to, because you said that this kind of mutation will, kids wouldn't be having HIV when they are born.
Mm-hmm.
So does that mean they will be also immunized, or they will not get HIV later in life, or this purpose was only for being born, then later in life that could happen?
So that's another issue with the study, is that if they had used current practices, like they have clinical practices of sperm washing with dads who have HIV so that the babies don't get HIV. But this study was so that they would be intrinsically resistant to it. So throughout their lives, they would not get HIV. Through sexual transmission, through blood transmission, like they wouldn't. So obviously we don't know if that worked, but that was the goal.
Yeah, because if that is the case, that like they can get the HIV later in life, what is the purpose of the whole study? Getting to all of this risk just to have them born without HIV, where you can do that in another way without any gene editing. So
Exactly.
I mean, it's somehow it's misleading, as you said, and I think there is no way for this to be ethical, as he said. Like, this is a complete playing with human genome, and they have got those babies to life with a misleading study and ethical reports that wasn't true. So I mean, I'm glad that he got this in court, and he got guilty for this, because I cannot believe that he could jump all to that point without doing that ethically.
Yeah, it's quite shocking that this happened, but
Yeah.
it's also crazy because it's very likely that things like this are still happening and will continue to happen, because all of this was obviously done without any like legal permission.
I was even thinking, like, if that happens through a university, how will that get funded and get work through all of that steps without being ethically in reality?
Yeah. So in this study, they said that he did it with 2 mothers. So one mother had the twins, and then the second mother recently had a child, but because everything's fully anonymous, we're not actually going to know what's going on with them. But yes, I do think I agree, it's extremely irresponsible and unethical.
I don't. I have such a hard time with this case, because when I think about what his actual intent was, I don't see an ethical issue with it. Like, the intent to have people that are resistant to HIV, I think is actually a really good thing to try to do. It's just the way he did it was unethical. So in that way, it puts like this really bad light on it. But if you actually look at what he was doing, it's hard to be like, oh, that's such a horrible thing. He didn't want these babies to ever get HIV. So it's like, I have a hard time with this case, because obviously he needed to be like reprimanded for what he did and stuff. But it's also like, I don't know, kind of amazing that it did work, and that we know that it now works in humans, and we kind of have proof of it. So again, it's like that going against what you're supposed to do is bad, but it did further science. So it's like, again, I don't know.
Yeah, I just had a quick comment, because I kind of feel, it's a bit of a gray area, which is why it's so interesting to talk about. When you mentioned, Ashley, about how his intent was to help others, there's a quote, I don't remember where I heard it, but it was through some like work training session about either ethics or something like that. And they said it's not always about like the intent, it's kind of the impact that you have as well. And kind of going back to what was mentioned earlier, I think on this slide is, he wasn't targeting those mutations directly, he was targeting genes around it. And then we don't know what the actual impact is on them either, because they have to remain anonymous. But on the flip side, I think when we had a conversation about this a couple classes ago about how you see scientists who have challenged and played around in the gray area a lot, and that has kind of pushed forward certain things within this realm, which I don't know a lot about, to be honest, and I think this might be one of them, but it'll be interesting to see kind of what the changes are after this, because we see, like I've been seeing a lot of articles about gene editing and how to navigate the ethical space regarding that too, but definitely I think it'll be interesting to keep an eye on it.
Yeah, the only thing I want to add, I had a similar comment to Leanne's comment actually, just about like intent versus the effect or like the consequence on the children. I think that the intentions may have been well intended, but what he was doing, like honestly, his staged intent may have been more of a facade for what he actually wanted to do. That's kind of where I look at it from, but I think that it would be really great if we did that sort of research ethically somehow and found a solution that could minimize, or I guess destroy, the risk of having HIV in the future.
Do you think that we see this case that is very complicated to analyze, but in many cases you need a precedent? Maybe this is the precedent that we needed to advance, just as you said, advance forward in this particular approach to curing diseases in the future. It was improperly done, it was sketchy, he was not transparent, and probably we needed more research done in animal models before doing such a big jump. But because he has done the jump, maybe we can begin using research in a more directed way to use this CRISPR technique to target very, very specifically some genes that can have a positive effect in the health, in the lifetime health, not only in the momentary health, but in the lifetime health of individuals. I don't think personally that we were there at the time he did it, but I don't know, at times some rogue scientist can show that we need to move in that direction. Do you think that we need to move in this direction right now, or that we're not ready?
Okay, well, I agree with a lot of things that are being said. This is the same, like I have the same issue when I think about this. Yes, every major breakthrough or every kind of advancement in a field is led by someone doing something crazy at their own risk. Like he said he was fully willing to take responsibility for what he's done if people want to punish him, which, okay, I guess it's admirable. I also guess his intent is admirable in that he wanted to not allow these children to suffer from having HIV for the rest of their lives. Yeah, I don't know. I find it really hard to kind of figure out exactly how I feel about these, because everyone agrees that it was unethical and too early. But when it comes down to the what actually happened, what the actual study that he was pursuing is, it gets tricky. Will gene edited babies ever be commonplace in the next like 5 decades? Will it be okay to just make sure, just have a checklist of like specific diseases that we know where they're located in our genome, and just say, okay, they're not going to have any of these, and it's just like a panel of immunizations, embryonic immunizations. So yeah, it's interesting and confusing to talk about.
This also opens up another can of worms, in that if we begin modifying humans in such a way and making better humans, they would discriminate unedited, non-edited humans, or I don't know. Anyway, sorry, I think I caught Ashley.
Sorry, I just want to like talk a little bit more because everyone was kind of replying to me, in that like everyone seems to vilify his intent, and I understand that kind of idea from people, but like this is also like the field that like I am in, and I love this field, and this like IVF and all this kind of stuff is like what I look at, what I study all the time, and like this is where so, but when I look at it, I see someone who had a good intent, and I don't know if we should vilify it and say he had some sort of bad intent or whatever. And a lot of these issues that were faced, had it been a peer-reviewed study, had he gotten the ethical like approval of it, a lot of these issues wouldn't have happened, because someone would have told him or caught and said that you can't implant these until like the Mosaicism is gone, or you have to target this specific mutation, or you have to test them and figure out that there's no off targets or whatever. And a whole group would decide that, and then it would be able to go through. That wouldn't have happened because they would have rejected him right away, because as a group, we don't think we're ready. So, but like, it's the way he did it that's the problem, not in my mind what he did. And he just did it wrong because he was doing it by himself. And every researcher makes mistakes when we're doing stuff by ourselves. That's why we have other people check our work. And that's why we have ethical boards that analyze what we do and figure out if this is okay. So because he skipped all of those steps that make all of these problems actually did exist. So it's not, I don't condone what he did at all, but like, I don't know if we should vilify his message because he was like, I don't know, over excited about what he did, because that's what all researchers do. We all want to go for stuff so hard. He just didn't have, I guess, the thought to like take a second and analyze more what he's doing, which is a mistake on his part, and that's why he's paying for it. But it's still I don't know.
So I think we all understand what you're trying to say. And I think we all feel a pretty similar way about it. It's difficult. I want to actually give you another example that was peer-reviewed, and it's not humans, so maybe it will be a bit easier to talk about. Okay, so this is actually a gene-edited and cloned group of monkeys. So macaque monkeys are really commonly used in neuroscience labs. So they wanted to make a specific type of, a specific phenotype of a disease. So in this one, they knocked out the BMAL1 gene, which is partially responsible for the circadian rhythm, so the sleep-wake cycle. And they chose the monkey that was exhibiting the most severe disease phenotypes and cloned it. So the one that was displaying the most severe depression, anxiousness, and schizophrenic behavior. So they cloned that. They made, I think, 5 or 6 clones of that monkey. There's a video here that you can watch later when this presentation will be up on eClass. It's pretty sad. It's kind of disturbing. There's like a bunch of baby monkeys. They're clearly distressed. They have really weird erratic behavior because they're not, they're not cloned from healthy monkeys. So the researchers defended this because they stated that this could actually reduce the total number of monkeys used in labs, because you can create the perfect custom model organism for what you're studying. Then you don't have to go through generational cycles of breeding the ideal disease model of the specific thing you're studying. You can just create them. But at the same time, these monkeys are being seen as merely tools to test things on. And in this case, they were just created to see what would happen. There was not really a purpose. They're just like, can we do this? What's going to happen? So yeah, does everyone feel the same way about this? Is it more okay because it's monkeys and peer-reviewed, or yeah, how do you feel about this?
For myself, I am also feeling against it, to be honest. Like, even if that's with animals, that doesn't mean we can just mess with it and see how would it go. In the end, like, this could be, I see that like trying to create a new brand of animals or new brands of humans that may we not control afterwards. So I still feeling against this one. And I don't think it would be an easy step to create this kind of a new brand of even human or animals, because even on the level of cell cultures or, small animals, even with breeding, in the end, we cannot get a perfect gene typing cell or animal, because cells are different. Like, we cannot even understand how does it, I wouldn't say behave, but I mean, how would it work as a cell completely? Because in the end, there are many areas that we don't still understand. So that's why we're still far away of doing such thing. So, maybe we feel more angry for humans because, if those little, gene edited babies grown up, I believe they would have a right to sue these researchers, because they have been, created. They haven't come to the world with the natural way humans know. And if they know that, and they should know later in life that they can't sue this person if they got any, uh, anything that's over control or something that couldn't be dealt with because the way they got to life. So I feel like this is actually my reaction. Still, we are far away of doing such thing, even for animals.
Mm-hmm.
Yeah.
I want to add up to this, and it's true that whenever you modify one gene in cells, Hadija and I, we work with cells. Sometimes you find an unintended side effect. You were like, oh my god, I wasn't even counting on measuring this other effect, but look at how interesting it is. And in cell culture, it's just interesting. In animals, it's also interesting. For instance, we know of a particular, whenever we downregulate a particular gene, the mouse gets better hair. And that's something that were not counting on. And, these are sometimes good outcomes, right? It's like, oh, wow, this mouse has a fabulous hairstyle. But we don't know yet if there are interactions, and there's evidence points that there are many interactions in between several genes. What happens when one of those genes is just deleted, or I don't know, what are the secondary effects, is something that needs to be studied. What is the best model to do it? I don't know. Maybe quantum computers, they can do it, in a virtual world, and then, that's okay. However, I think that in the end, in the future, we will have humans that are modified for good purposes. We don't want cancer around. It's horrible going through the whole process as a family is a mess. What if we can create a human without cancer? That's a good overall purpose. And I understand what Ashley is saying. This man or at least he seems to have a good intention, right? To get to that point. Um, and it's very subjective, I think, our opinion on him. He has a good point, but we may not be there yet. If doing it with animals is okay, I feel just like Hadija. I, I mean, these are, and I'm biased. I have a pet, you know this, and I'm like, no, you don't touch my baby bird because he's like my baby. So you know, it's difficult. And just by the way, Taryn has commented on the chat, Ishita, and I don't know if you would like to read that and
Sure. Yeah, so Taryn says, I have a lot of feelings about this one. I can't help but think that the sticky part here is the fact that monkeys are primates. This really begs us to ask where we draw the line ethically. I'm inclined to say that this is ultimately highly context dependent. What is being studied, what alternatives are available, how many organisms are needed, etc. Yeah, so that's her comment. She doesn't have a microphone on her computer, so that's the reason that we do this
and
reading according to Taryn. Yeah.
Just touching base on what alternatives are available. There are new developments on artificial organs, because sometimes if you study a particular cell in a petri dish, is it really what is happening in the human body? Because we are a collection of different cells working all together. So having an organ would be more similar to the real thing, right? And now they are developing these hearts in this bioreactor, which is fabulous to see. So there are alternatives on the becoming path, right? We are getting there, and yeah, that's the only thing I had to say. That we're also developing good alternatives to study these kinds of approaches like CRISPR.
To add to the organ thing, though, that is a lot better than just looking at cells, but the body is an integrative process that a lot of things like start in the hypothalamus, go to the pituitary, and then go to our organs. So you're still leaving that kind of part out. And with the, sorry, I had a lot of, with the talking about how if this were to happen and like growing up, you would sue the experimenter because they altered something in you. So does that mean if a child, like if a mother decides to go under IVF therapy and something goes wrong with that process, the child grows up and they have some sort of illness because they went through the IVF process, because there is environmental factors that can alter it? Does that mean they should then sue the people who performed the IVF procedure on them? And monkey, so the one thing about this particular study is that like it also kind of goes to if you think it's ethical to breed monkeys that have these issues, because that's normally what they're doing to study them, is they're breeding monkeys that have schizophrenia. I don't remember what the actual issues were, but they're breeding them and doing multiple rounds of it, of different generations that have these issues as well. So is that really that different than just creating one right away that has this issue and not going through multiple cycles of it? So that positive is where I can kind of see that it's good. But because we also do a lot worse things to rodents as well, but it's ethical because we decided it was ethical to do this to a rat, even though a rat is very intelligent as well, or pigs, or whatever animal we do this to, we are harming them in some way, but we decided as an ethical committee that this is ethical, as long as we do not as much harm as we don't do more harm than is needed, but to study these things, we're still causing them harm in the first place. So it's a whole encompassing thing of like, is research worth harming animals to get answers?
Yeah.
No matter the animal.
I had a whole slide of questions to talk about, but I think you guys touched on basically everything, which is really great. Like I'm really enjoying this discussion. But quickly before moving on, I just wanted to talk about this shift in the framework of what we see as medicine, from what we saw in the past examples of Lulu and Nana. So the traditional model of medicine is that a patient has an issue, the patient comes to the doctor to get treated, and the aim of treatment is to have them no longer be a patient, to kind of give them enough therapy that they no longer are dealing with the issue. But with cases like the HIV genetic modification to prevent HIV, not just at birth, but for the rest of your life, that's something different, right? So this is like a patient that could have been born without HIV, and they're actually being enhanced, and we're not sure what the consequences are going to be. So this is also like when we talk about in class modifications to healthy people, like we talk about adding technology or like cyborg humans. This is kind of similar in a way. Some people think that it might be more similar to immunizations, and some people think it might be more similar to enhancements if you're actually changing the genetic makeup of the baby. So I just wanted to point out this kind of societal conceptual shift that we talk about when we're talking about the exponential future, and that distant future might actually be kind of starting, the rumblings might be starting to happen right now. Yeah, I think like, okay, I have another section about intelligence and consciousness, but if anyone has like any last comments on this, speak now, and then we can move on to the next section. Okay, I'll move on to the next one. The next part that we're talking about is intelligence and consciousness. This is, I mean, it's unrelated to the first section, but I just think it's extremely interesting. It's something that I'm very interested in. I'm in neuroscience right now, so this is something that I think about a lot. And the fact that a lot of the time when we're talking about artificial intelligence, we just tend to assume that at some certain point of intelligence, machines will just become conscious. Like they're so smart that they have become conscious. But I think that it's important to kind of draw a distinction between intelligence and consciousness, and that one does not necessitate the other, I believe. I mean, I know this is something that is debated by scientists and philosophers. So here's, I'll try to present an unbiased view and then we can talk about it. So when we're talking about intelligence versus consciousness, the most prevailing theory right now is that intelligence deals more with the external world, how we deal with everything around us, whereas consciousness deals with what's inside of us. So we'll talk about the external world first. So human intelligence is defined as a mental quality that consists of the ability to learn, adapt to new situations, understand and handle abstract concepts, and use knowledge to manipulate one's environment. So a lot of this, the traditional theories of human intelligence are just based on our ability to respond to things, to react to things outside of us. So based on this definition, it seems pretty clear that machines are already here, right? They can adapt and respond to the external world. So I just wanted to give you a concrete example of this because it's cute. It has clinical applications and it's really interesting. So this is the Paro seal. It's being used for quite a few years as a clinical device to help patients who have dementia or Alzheimer's, to provide socialization and just kind of improve the quality of life for these patients that deal with kind of social isolation and things like that. So this is used pretty regularly in like old folks' homes and in patients with neurodegenerative diseases. It has a bunch of different sensors. It can tell when you're talking to it. It can tell its name. It can tell when you're saying something happy to it or when you're angry at it. It can tell which way you're holding it. Like if you're holding it like a baby, it has like a posture sensor when you pet it. I thought this was really interesting. If you stroke it every time you touch it, Paro will remember your previous action and repeat that action to be stroked. So it wants to be stroked. If you hit it, it remembers its previous actions and tries not to do that action. So this is, I think this fits really well into that definition of human intelligence that we saw. It can react to and adapt to the external world. So if this is the bar for artificial intelligence, we've obviously already reached it, which is why I want to draw a distinction between that and consciousness, which is a lot of what we're talking about when we talk about like the future, like robots that we can hang out with type of artificial intelligence. So this is a pretty busy diagram, but all I wanted to kind of point out with it, this is the behavioral generation system of the Paro seal. But this is also very similar to how we generate behavior. We see the stimulation, we examine it, and based on our previous experiences, we decide how to respond to it. So everything, all of our relationships with the external world are based on predictive patterns. Everything is our best guess of what we should do or what the right response is. And I know this is really abstract, so I wanted to show you an example because this isn't just how we're dealing with social relationships and things like that. This is also how we deal with even things like our own body. So I will see if this plays, otherwise I have it open. Basically, this study uses a rubber hand placed in front of a person. Can you guys see this still screen right now? So
yeah, it's a rubber hand, yes, because I got a little bit freaked out.
Yeah. It's a rubber hand, and someone places their rubber hand in front of you, in front of the participant, and their other hand is placed beside them, but they can't see it. It's blocked off. And both the rubber hand and their actual hand are stimulated in the same way. So in this study, they take a paintbrush and stroke it on the same finger at the same time for the fake hand and the real hand. So you'll see how this participant's brain begins to start, because it resembles a human hand and it's about the same place where his hand should be and feels the same things his hand should feel. His brain is fooled into thinking that this is a pretty good prediction of where his actual hand is. So I will try playing this. Can you guys see anything right now? Okay, I will reshare. All right. You guys can see the video now? Perfect. So, all right. Okay, so it's a really simple experiment, but it works. And I think it shows you how prediction-based our intelligence really is. It's really easy to fool our brain into thinking something that's not actually our limb is our limb, because based on all the other input it's getting, it's close enough. So moving on to the internal world of, or the self, as compared to the external world, which we know can be easily replicated in machines. This part is where it gets really tricky, and we don't really have any answers to right now. But basically, there's two camps of thought. In that the first one thinks that consciousness is deeply tied to biological mechanisms. So a lot of scientists think that consciousness necessitates our biological being. So it's based on our organ systems, how we regulate our body, things like that. Whereas other people, other scientists think it's a byproduct of specific regions of the brain being stimulated. So that's the picture I have over here. So there's this posterior hot zone that is extremely conscious, extremely active during a conscious experience. And lots of other parts of the brain, including the anterior part of the cortex, and then also the cerebellum, where people who have had injuries there or who were born without a cerebellum at all can still have the same conscious experiences as we can. So people have begun to isolate areas that have nothing to do with consciousness, and this posterior hot zone has been identified. So if it's just a series of connections in a specific part of the brain, that makes it much more easy to imagine that this can be isolated and replicated in machines. Whereas if it's something that is actually connected to our biological mechanisms and can't be separated from it, obviously that would be much harder to replicate in machines. So this is my last slide, because there's not really any kind of concrete evidence about any of this. So I just wanted to kind of talk about it because I think it's really interesting, and I was curious to hear your thoughts on it as well. Does anyone have any burning questions or comments?
That's I don't know if in our lifetime someone will define what consciousness is. For instance, if you have very vivid dreams, how do you differentiate those dreams from the reality, which is now, right? Is this a dream or is it not? And so yeah, I like that we think of this, and also I liked that you introduced the similarities between these the Paro seal with our interpretation of the world, because we are a collection of sensors, really, that are getting all this input from around us, and then, as you said, based on previous data, we get better at predicting, oh, probably, this is the proper way in which I should answer this input, right? So it's true, the machines are getting there, but what makes us different is consciousness, and what is consciousness is still not defined. So I mean, it's I guess maybe something that once defined, we will try to replicate in machines, and maybe that will be the day in which robots will become humanized. I don't know. Yeah.
There are some things specific to kind of a strategy of the students in this course. So it seems like politically, transhumanism is in kind of a mess currently and has been for some time. And you can just simplistically think that a group of bright young students could sort of fix that, right? And so this is one of the hot areas. So David Pierce believes that the binding problem, as he calls it, the difference between the physiology of what's going on in the brain and actual consciousness is a problem that won't be solved, for decades. And then in our quantum quantum biology lectures, there's a slide that Jack has been using that basically says the same thing, that, you know, we think that we're getting close to this, but we really have no idea what consciousness is. So maybe if you think like Taryn and Leanne at the moment are thinking that they would try to do something in this area, maybe the students in this course would eventually make big strides, big progress in this area that older people in the transhumanism area don't seem. Okay, let's see what Taryn said. Okay. Taryn says, I've seen consciousness referred to as a spandrel of intelligence, meaning that could be an evolutionary byproduct of developing highly complex brains, more intelligence. I don't think that is a completely satisfying answer, but interesting in that it can point to other beings developing consciousness as well. Yeah, and so.
But then are humans the only ones capable of
No,
being
no,
conscious, or?
no, no one's saying that. But I mean, obviously, your lovebird is conscious, right? Maybe more than you are, right? It's a question: which of the two of you are more conscious, right? Yeah, but there's a kind of knee-jerk reaction among a lot of people interested in artificial intelligence that, oh, I can't really explain why, but when things get intelligent enough, they will be conscious. You know, don't worry about the reasons, it'll just happen, right? So, but we really don't have any evidence for that at all. Yeah, so rather than it seeming terribly remote, you know, you guys, after all, you're the only group of students with cardboard cutouts. You can maybe solve this, right? So you can make a situation in the future where we understand this much better than we do today. So that is a possible way in which you could make a difference in the world. Yeah, just trying to inspire you a little bit to think about what could happen here. Yeah.
Does anyone else have any thoughts before we wrap up?
Yeah, we've got 3 minutes until the official end of the teaching session. So any other thoughts? There's, you know, a difference in how Zoom videos end up. In the beginning, the video recording would show what you're all seeing now, would show like multiple pictures of the people taking part and the slide set. But I guess because in many parts of the world, the pictures of the people are really uninteresting or possibly disturbing, they decided to change it. So when somebody's doing screen sharing, you only see that slide, right? You don't see the other stuff. And so it's a kind of dramatic difference. I don't know exactly which version of Zoom changed that or whatever, but so what it means is the experience of actually being here is a little bit different from the experience of the video. Like you'll only see Ishita on the video when she stops screen sharing. So, right, so, every now and then she's stopped and started. You'll see her then, but you won't see her while the actual slides are up.
Okay, if no one has any more comments or questions, I just wanted to say thank you so much. You guys were a very involved class, so it was really fun for me as well. So thanks so much for sharing your thoughts. And yeah, you can, if you have questions or if you want to talk to me more about any of this stuff, I find it very interesting to talk about, so you can always email me. Yeah, thank you.
Great. Thank you very much for a great teaching session. Okay, guys, so the next 3 times are ethics. So Dr. Van Manen, the first and third time. Yeah, so anyway, that's the immediate plan from this time onward. Okay, so we'll see you next week.
Thank you.